Welcome to Introduction to Ecology, Lesson 16. Today we're moving on to a new type of interaction, and that is competition. Now, unlike the previous two lectures where we looked at predation and parasitism, competition is a non-trophic interaction. It doesn't involve feeding of one species on the other. Instead, competition is an interaction between two or more species where all species are negatively affected because they share use of a particular resource or a set of resources that limit their ability to grow, reproduce, or survive. Sometimes that resource might be food, but sometimes it might not be. Broadly speaking, we can divide competition into two classes: intraspecific competition and interspecific competition. Intraspecific competition is competition between individuals of the same species. So on the right, you see that figure of the two rams butting heads. They're competing for mates, but they are the same species. So this is intraspecific competition. Interspecific competition is competition between individuals of different species. So on the left, you see hyenas and a lion competing for a food resource. So again, two different species competing for that shared resource. Today's lecture is primarily going to focus on interspecific competition because in this particular unit, we're interested in species interactions between different species. And so interspecific competition is the form of competition most relevant. So what kinds of things do species compete for? Well, they can compete for any resource or any component of their environment. This might be food, it might be water, those are pretty obvious ones, but if you're a plant, sometimes the most important resource is light. That's how you make your food. And for both plants and animals, sometimes a very important resource is just space. You need physical space to build a nest. You need physical space to be in a position where you can absorb light that's coming to the earth. So again, these are all different components in the environment that are required by species, and each one of those is a resource that can be limiting, that can be a resource that different species compete over. In a previous lecture, we talked about ecological niches, and we introduced the idea of the fundamental niche and the realized niche. So the fundamental niche is the full set of resources, along with biotic and abiotic requirements, that define where a species can live. But then the realized niche is the restricted set of resources, along with biotic and abiotic requirements, that define where the species does live. So the fundamental niche is basically everywhere you could expect to find that organism based on the conditions, things like the salinity, the temperature, the amount of rainfall, the precipitation, that sort of thing, the tide level. But then it doesn't necessarily live in all those places, and frequently it doesn't live in those places because of biotic interactions. And one of the most important biotic interactions is interspecific competition. We saw that with Connell's barnacle, where the brown barnacle should be able to live everywhere from the very low tide zone all the way up to the high tide zone, just based on what it can tolerate in terms of abiotic conditions. But it's actually outcompeted in those lower tidal zones by the blue barnacle that's a stronger competitor but can't actually live all the way up to the top. And so again, competition is one of the big factors in reducing the size of a species' realized niche relative to the fundamental niche. So there's a couple of different types of interspecific competition. One is what we call exploitation competition. So this uses the word exploit, so not surprisingly, this is a type of competition where individuals of different species exploit the resource, and as a result, it becomes limiting. So this is basically when individuals reduce the supply of a shared resource as they use it, and so species that come in later to try and use that resource, they just can't get at it because it's already been removed. A good example is the amount of light in a rainforest. So remember we talked about the different rainforest levels, that emergent layer, the canopy layer, the understory, all the way down to the forest floor. Well, not surprisingly, as light comes from outer space, it hits the emergent layer first, and some of it is soaked up by the leaves in the emergent layer. And that reduces the amount of light penetrating down further to the canopy. The canopy then takes its own share of the light, and then that means even less light reaches the understory all the way down through to the bottom. And so you have to have very shade tolerant or plants that can deal with not a lot of sunlight at the bottom, because that sunlight has been reduced as it goes through and hits the other layers, the other species that are up higher first. Another type of competition is interference competition. So this is sort of the opposite. This is where it's not so much using up a resource so there's none left for the next guy in line. This is where there's a direct interference with the ability of competitors to use that resource. Typically this is warfare. So a good example is with ants. Here we see two different species of ants that are fighting one another for food. And again, it's not so much that one ant is coming in and using that resource first, and then there's none left for the next guy to arrive. Instead, they're directly fighting over it. They're pulling that resource apart, and they're taking each other's legs off, and they're killing the members of the other colony. That's interference competition. One very specific form of interference competition is what we call allelopathy, and this is something that happens in plants. This is where individuals of one species release toxins that harm the other species. So again, very important in certain plants, and especially even in some crop systems. Unfortunately, it's been hard to demonstrate in nature too much. We believe it happens, we believe it's quite prevalent, but we don't necessarily know how much. But we're finding new genetic variants that we can control toxin production on, and that's actually allowing us to test the effects of this toxin production on competition between plants. Again, this is a form of interference competition because it's not that one plant is using the light before the next one gets to it. Instead, the plants are actively trying to kill each other. And so that's why it's interference, not exploitation. All right, so what is the most important limiting resource? That's ultimately what's going to restrict species. It's ultimately what they're going to compete over. Well, frequently what is limiting depends on where you are. And so as a result, competition for the limiting resource can change depending on conditions. Regardless, whatever is most limiting, that's where your competition is going to be most intense. So let's take a look at this example. In this example, I have some trees with some nesting sites. Nesting sites could be a limiting resource. And I have a mouse, which is prey. And again, the prey, the amount of food, could be limiting. And let's suppose I have two different owl species. Well, in this example, I have a lot of nesting sites, so they might be limiting, but they're not the most limiting thing. But I don't have a lot of prey. And so these two owl species are going to compete most intensely for that prey. But let's look in a different region of the forest. Maybe in this region I have far fewer trees and far fewer nesting sites, but because it's kind of field-like, I've got a lot of mice running around. It's a great habitat for mice. Well, in this case, maybe the food, the prey, is not the most limiting resource anymore. Instead, it's finding those nest sites. And so in this case, the two owl species are not going to compete as intensely for the mice. Instead, they're going to compete most intensely for the tree. And so again, exactly what the limiting resource is and what the species are competing most intensely for depends on what's available. And that can change from one place to another. It can change from one time to another. Typically in competition between species, there are winners and there are losers. So competition usually reduces the resources available to all the species that are competing, but typically one of the species is more harmed than the other. And in some cases, one of the species is so harmed, is so limited by the other competitor, the other competing species, that it's driven to extinction. And a recent paper with a good example suggests that humans actually outcompeted Neanderthals, and that's why Neanderthals went extinct. It actually had to do with humans being the stronger competitor and just getting the limiting resources that both needed to survive better than the Neanderthals, and as a result, the Neanderthals went extinct. Now, what you'll notice in this write-up here, the authors of this study actually used a Locke-Volterra model to prove or to offer sort of evidence that humans drove Neanderthals extinct. We're going to talk about, we've already talked about one type of Locke-Volterra model. We've talked about Locke-Volterra predator-prey. This is a different model. This is a Locke-Volterra competition model. Same people devised it, but we'll talk about that a little later on in today's class and then in the next class. And so you'll see how these simple models that we're actually talking about can be used to study new systems and find new things out, even about humans. So I said most times both competing species are harmed by the competition. Nobody's a total winner, but there are sort of better species who do better and species who do poorly. That's not 100% true. Sometimes in some competitive interactions, one species is basically not harmed at all. The effect of the competitor is zero or almost zero. When this is the case, we call it amensalism. And one example of amensalism would be small woody plants growing beneath the sort of emergent layer in the rainforest. The emergent layer in the rainforest doesn't even really know that these tiny plants are below it. But the tiny plants definitely know that there's a lot of leaves above them taking the sunlight before it gets to them. So again, in this case, the big emergent trees don't really feel any strong competition from the tiny little plants at their feet, but the tiny little plants feel a lot of competition from the big trees. So that's an example of amensalism for the emergent layer, for the big trees. They don't feel that. Often we think of competition being something that happens between similar species. And that makes sense. Similar species share a lot of the same resources, and so as a result, they're likely to compete for those resources. But sometimes very distantly related species can actually be strong competitors. The only requirement for competition is that they use the same resource. They don't have to be similar. They don't have to be sort of evolved from sort of the same lineage. They can actually be very, very different animals, very, very different plants. And here's a great example. So there was an experiment done in the desert where we had some rats and we had some ants. And both of these species used seeds as a resource. It was one of the most limiting resources for these species in this environment. So what scientists did was they removed ants from some plots, and they did that with an insecticide. And so in those plots, you only had the kangaroo rats. Then in other plots, they removed rodents, and they did that with some caging that was too small to let the rodents in, but the ants could freely wander in and out of the cage openings. And then there were other plots where they removed both ants and rodents. In those plots, they used pesticide and the cage. And then there were some plots where they removed neither. And then what they did was they compared these different treatments to understand how the ants affected the rodents, how the rodents affected the ants, and how both of these affected seed availability, which they hypothesized was the limiting resource. So if they compared the plot where both ants and kangaroo rats lived to the plot where just the kangaroo rats lived, what they found is that there were 18% more kangaroo rats, 18% more rodents, and actually 24% more biomass. The rodents themselves were bigger as well. And there weren't just more of them, they were bigger when competition with the ants was removed. Then when they compared the plot with ants and rodents to the plot with just ants, where they'd removed the rodents, they actually found that there were 71% more ant colonies when the rodents weren't there. Then when they looked at the amount of seeds that were left over, and they compared the plots with just the mice, or just the rats, just the ants, and then the rodents and the ants, the rats and the ants, they found that the same number of seeds were in all of these plots. So it didn't matter whether it was just rats or just ants or a mixture of the two. In all cases, the same number of seeds were being removed and the same number of seeds were left. And the number of seeds left was much, much less than the number of seeds left in the plots where neither the ants nor the rats were allowed to get into. And so what that told them was that ants reduce seeds available to rodents. And as a result, the rodent population is limited by the presence of these seed harvesting ants. At the same time, rodents clearly reduce the seeds that are available to ants. And so the size of the ant population is limited when there are rats present. So that's an example where you have two quite distantly related species. You have ants and you have rats. One's a mammal, one's an insect, but because they both require seeds as a food source, they compete directly. We have other cases of competition between more similar species, things like two ants. Regardless of whether we're looking at different species, very, very distantly related species or very closely related species, there are sort of two possible outcomes of interspecific competition. The one outcome is what we call competitive exclusion. So in this case, the dominant species actually prevents the other species, the less dominant species, from using the resources that it needs, and as a result, that inferior species, that weaker competitor, goes extinct. It may not go extinct across the entire world because, again, conditions can change from one place to another, one time to another, but it will go locally extinct. It will go extinct in that region where the stronger competitor exists. And so, a good example in our neighborhood, I tried to do a lab last year in the experimental forest, and it was a lab where we were going to look at how rapidly different ant species discovered food. And having done a lot of research in the southeast, I assumed that the seed-dispersing Aphenogaster ant would be prevalent everywhere and would be sort of really fast at finding the food, and then some other species would come in. And so I sent my students out in the field, and I didn't go with them the first lab session because the bus didn't have room for me. And they all came back and they were like, we didn't find a single Aphenogaster. In fact, we only found one species of ant. We only found needle ants. And I said, no, that's not possible. That's not possible. Aphenogaster is always the first to the bait. There's no way you didn't have Aphenogaster. Maybe you just didn't key it out right. And so I went with the second group into the field, and I couldn't believe it. The only thing I found on those baits was needle ants. And needle ants are an invasive species. They come from Asia. And in fact, what had happened, I know that historically we had Aphenogaster in these woods, and they were the most dominant ants. They were widespread throughout the entire southeast. In fact, they're widespread throughout the entire east coast. But what has happened in the experimental forest is that these needle ants have competitively extinguished Aphenogaster. And so now there are no more Aphenogaster, or almost no more Aphenogaster. I have not seen one in my entire time. So that's one option. Now, fortunately, or unfortunately, depending on how you feel about ants, the needle ants have not excluded all ant species from the experimental forest. There are still other ant species, and those ant species do compete with the needle ants for certain food resources. One good example is carpenter ants. They're still quite prevalent in the experimental forest, and so we did find a couple of them on our baits. So that's the other possibility, and that's competitive coexistence, where two species are able to coexist despite the fact that they share limiting resources. And this has to be coexistence that occurs indefinitely. It can't be coexisting for a few years until the one of them gets wiped out. That's competitive exclusion. When I say coexistence, I mean they live together forever and ever and ever and ever. All right. So let's go back to competitive exclusion. The competitive exclusion principle states that two species that use a limiting resource in exactly the same way cannot coexist indefinitely. And so basically, if there's no difference at all in how they use the resources in their environment, one species will always be just slightly stronger than the other. And even if it's a fraction of a hair stronger than the other species, over time it will drive the other species extinct. And so that's probably what's going on in the experimental forest. If we look at Aphenogaster and we look at needle ants, they have almost 100% overlapping requirements in terms of their resource use. So they're both primarily limited by food availability. They're not limited by nest sites. They nest in the ground and the leaf litter. There's plenty of leaf litter and ground available. They're not limited by water. Most ants are pretty hardy to dry conditions. So the main thing that's limiting both of these species is food. And as a result, they're competing for that same limiting resource very strongly. Even worse, they both eat almost identically the same food, and that is pretty much everything they can find. They eat dead animals, they eat any dead insects they can find, they eat any live insects they figure they can attack. They both find food rapidly compared to other species. And unfortunately, because they have sort of that same strategy, they eat the same foods, they are limited by the same things, the needle ant is slightly, or actually quite a bit, more aggressive than Aphenogaster, than the seed-dispersing ant, the native ant, and as a result, that's why these needle ants are driving the Aphenogaster extinct. It's the competitive exclusion principle. And in fact, this should not have shocked me. I knew that there were needle ants in this area even before I sent my students into the experimental forest, and I also knew in a previous study in another region in the southeast that researchers had found 96% reduction in the Aphenogaster populations in places where the needle ant had been introduced. And so it should not have been surprising to me when my students came back and said, we found a lot of needle ants and no Aphenogaster. I should have known that was what happened, because we have demonstrated a competitive exclusion of the Aphenogaster by the needle ants in multiple different places. Now the experimental forest. All right, so competitive exclusion is sort of the one way things can go, but sometimes competitors coexist. And the reason that competitors can coexist is because they do what's called resource partitioning or niche partitioning. What this means is that there are differences, slight differences, in how the species use limiting resources. And basically those slight differences in how they use the resources, in what their strengths and weaknesses are, and exactly which resources they're using when, all of that allows them to differentiate from one another and sort of fill these distinct niches and coexist. So a great example is with mosquitoes living in things like bromeliads, pineapple stuff that looks like pineapples. So bromeliads have water inside that middle part of their leaves, and that's actually great mosquito larval habitat. And if you go into these bromelid pools of water, you can actually find multiple different species of mosquitoes. And there was a study that looked at 4 different species of mosquitoes that appear to coexist across bromelid plants. There's 2 species of Wyomia, there's an Anopheles species and a Culex species. And so the question is, how can all of these larval mosquitoes coexist in these bromelids? Because they're all eating detritus, they're all eating sort of dead plant material that's in these pools of water in the bromelids. And so what scientists did was they looked at how the mosquitoes used the bromelids. And what they found was that different mosquito species preferred different sizes of bromelids. So you did find most of the mosquito species across most of the bromelids, but you found Wyomia melanopus primarily in your smaller bromelids, and you found Culex primarily in your bigger bromelids, and then you found the other 2 species mostly sort of at intermediate sized bromelids. They also found that the different mosquito species foraged at different depths within each bromelid. So these bromelids are like tubes of water, right? That central core makes a tube of water, and they found that the larvae of the different mosquitoes were eating at different heights in that water column. And so, for instance, the Anopheles was foraging at a pretty high depth near the surface, and then all the way down to one of the Wyomia species was foraging at a very low depth near the bottom of the bromelid. So you might look at these figures, you might say, yeah, but look at the Culex species and that first Wyomia species. Those are they're foraging at the same level in the bromelid, right? So they're kind of sharing the same resource in that water column in the middle. But if you actually look at those 2 species in terms of the sizes of bromelids that they use, they have the most different sized bromelids that they prefer. So Wyomia melanopus likes the smallest ones and Culex likes the biggest ones. So even though they forage at the same height in the water column, they're actually differentiated in terms of which size of plant they prefer. Then you might look at the Anopheles and Wyomia circumcincta, and you might say, oh, but they're in about the same size plants. They're in medium-sized bromelids. But then notice that they are actually foraging at the most distinct depths. So they're partitioning the depth along the water column most, and that allows them to exist in more similar sized plants. So again, these multiple mosquito species can coexist in bromelids because they partition both on plant size and on water depth within the column. And those slight differences in where they are and what plants they're choosing more than other plants allows them to coexist. A classic example of competitive coexistence is with warbler species. So these are MacArthur's warblers. They're all insectivorous. That means they eat insects, and they're not picky about what insects they eat. They'll eat any insect. So you might think, well, there's all these different warbler species. They're all kind of eating any insect they can get, right? That's kind of like the ants we talked about, where they're just eating whatever food they can get. You'd think there'd be intense competition between these warblers. And so for a long time, people wondered, well, how do they coexist? Why doesn't one of these warbler species outcompete all the others and drive them extinct? What they found is that each species uses a different part of the tree to forage for insects. And because they're they will use the entire tree, but because, one species mostly uses one area and another species mostly uses another area, those differences in where they prefer to forage actually allow them to coexist with one another. So sometimes competition can lead to extinction, sometimes competition can lead to coexistence, and sometimes competition actually leads to evolution. When that's the case, one thing that can happen is because species that are more different from one another can coexist, evolution can actually drive two species to differentiate. It can drive one to sort of go down one path and one to go down another path so that they can have niche partitioning, they can have resource partitioning that allows them to coexist so that neither one goes extinct. When this happens, we call it character displacement. So again, this is competition that causes the phenotypes of the competing species to evolve to become more different from one another over time. And one good example is with mud snails. So mud snails live in ponds and estuaries, and they basically feed on the sediment, the muddy stuff in there. And there are two species of mud snails that can occasionally occur in the same ponds. One is Hydrobia ventrosa, and the other is Hydrobia alba. And when these two species live alone in ponds, when only one species is found in the pond, then they're about the same size. But when the two species are put into a pond together, or when they coexist in certain areas, in certain regions they coexist in the same ponds, what you find is that the size of the snails diverges. And so the size of Hydrobia ventrosa gets smaller and smaller and smaller, and the size of Hydrobia alba gets larger and larger and larger. And what that allows them to do is to eat different sized particles in the sediment. The little snails tend to eat the smaller size, and the bigger snails tend to eat the bigger size, and as a result, they partition the sediment so that they can coexist. Contrast that with the medium-sized snails that can eat everything, but could not coexist with one another. And so natural selection often favors individuals of both species that allow them to partition the resources, that make them more different from one another, thereby decreasing the strength of competition between the two competing species. But that's not always the case. Depending on the species involved and the interaction involved, sometimes natural selection favors the opposite scenario. Rather than favoring competitive coexistence and pushing species towards coexistence, it can actually favor competitive exclusion, and it can drive the winner to be even more of a winner, so that that winner eventually destroys the other. And so natural selection can favor individuals that can outcompete the competitors and drive the competitors extinct. When that happens, we do not call it character displacement. That is not character displacement, is when the two species move further apart from each other so that they can coexist. When the final outcome is competitive exclusion, that is not character displacement. All right, so now we're going to talk about how we model competition. We talked about how we model population dynamics for single populations. That was the whole last unit. Now we're going to talk about how we model species interactions involving competition. And we're going to start with an equation that we already know. We're going to start with logistic growth. So again, logistic growth is a common form of density-dependent population growth. It's what we studied in the last unit. And basically with logistic growth, when you have low abundance of your species, the growth rate's really high and it grows up rapidly until resources become limiting. And then there's sort of not enough resources to go around, and so the birth rate of the species slows down and reaches some maximum sort of population size. And that maximum population size that the population reaches is what we call the carrying capacity. So basically that's how much resources available in the environment to allow that species to get to a particular population size. So the logistic growth deals with intra-specific competition, right? The competition among individuals of the same species competing for this limiting resource ultimately caps the population at the carrying capacity. So not surprisingly, when we want to talk about interspecific competition for a limiting resource, we start with this same basic framework. So just to go over it briefly, remember this is the differential form. You never actually had to use this. You kind of had to understand what the terms were and what they meant, but then we integrated it for any calculations you did, but you have seen this before. So on the left, we have the rate of change of the population through time, and that depends on the current population size. It depends on the per capita intrinsic rate of increase, or the population growth rate. and it depends on the carrying capacity. And remember, what we saw is that as the population size gets bigger and bigger, as n gets bigger and bigger, that n over k, that fraction, gets bigger and bigger and gets closer to 1, and so that whole bracket gets smaller and smaller and smaller until basically there's no, once n equals k, there is no change in the population size with time. It's reached that carrying capacity. It cannot grow any bigger. So that's logistic growth. And so, again, logistic growth describes intra-specific competition. And once you get to high population densities, there's less resource available to any one individual, and that reduces your population growth, either because species starve and die, or they just can't get enough food or resources to make new babies. And so here's an example with intra-specific competition. I've got two carpenter ants. They're fighting over a cookie, and one of them gets the cookie, and the other has no cookie to bring back to the colony, and so that colony is not going to do so well. It's not going to make so many babies. Very, very similar thing happens with interspecific competition, right? When another species is stealing the food, it's kind of the same as if your own species was stealing the food. There's just less available for you. There's less available for the focal species. And so just as if your own members of your own species were stealing that food, there's less for you, and as a result, you either starve or you don't make as many babies. There's less resources, and so the population growth rate goes down. But there's one difference. So, you can't compare apples to oranges. And so clearly this carpenter ant is going to take more of the cookie than this little winter ant here. He's going to steal the little piece of the cookie. And so what the winter ant needs to build his colony or her colony is actually smaller than what the carpenter ant needs to build her colony. And so basically the individual effects of interspecific competition may be more or less or the same as the effects of intraspecific competition, right? So individual for individual. If I compare one ant to one ant, then, if they're different species, the one ant of the different species may have a different effect than the one ant of the same species. It may have a greater effect than the ant of the same species. It may have a smaller effect of the ant of the same species, or it may have the same effect of the ant of the same species. And so we need to account for that when we're building a model based on logistic growth that captures both intra and interspecific competition. So here's what we come up with. This is what we call the Lotka-Volterra competition model. And it's, in this most basic form, we only look at two species, and we look at competition amongst these two species. And so it looks very, very much like the logistic growth equation, except that there's two equations. The first equation describes the growth rate of the first species, and the second equation describes the growth rate of the second species. But if I ignore that last term for a minute, what you'll notice is that both of these species, on their own, if the other species isn't around, both of them grow according to logistic growth. So again, if the other species isn't around, those terms disappear, and we have logistic growth. And so again, we have the rate of change of species 1, and that's dependent on the size of the species 1 population, its intrinsic rate of population growth, and its carrying capacity. And we have exactly the same thing for species 2. All right, but like we just saw on the last slide, just as intraspecific competition slows down the population growth rate, so too does interspecific competition, right? It doesn't really matter if your own species is stealing the food or other species is stealing the food's still gone, and so you can't breed as much and you die more. And so it doesn't really matter who's stealing the food. When the food's getting stolen, the growth rate of the population slows down. And so when we have two species in the area, we have to consider how much the individuals of the same species are suppressing population growth, but also how much individuals of the other species are suppressing population growth. But again, we can't compare individual for individual when we're comparing across species, because a different species may have a greater or a lesser effect than members of my own species on how much resources being removed, how much of the resources I need are being removed. So again, the effect of each individual of species 2 on survival and reproduction of species 1 may be more or less or the same as the effect of each individual of species 1. And so we capture that by including a parameter in these equations. And in the species 1 equation, we call that parameter alpha. When alpha is greater than 1, each individual of species 2 has a bigger effect than each individual of species 1 does on the species 1 growth rate, on the population growth rate of species 1. When alpha is less than 1, each individual of species 2 has a smaller effect than each individual of species 1 does on the species 1 population growth rate. And when alpha equals 1, then each individual of species 2 has exactly the same effect as each individual of species 1 does on the species 1 growth rate. Same kind of deal with species 2. Again, we have to realize that species 1 may have a smaller, larger, or equal effect on species 2 as species 2 does on itself. And so when beta is greater than 1, each individual of species 1 has a greater effect than each individual of species 2 does on the population growth rate of species 2. When beta is less than 1, each individual of species 1 has a smaller effect than each individual of species 2 on the species 2 population growth rate. And when beta equals 1, then it's basically the two are interchangeable. Each individual of species 1 has exactly the same effect as each individual of species 2 does on the species 2 population growth rate. All right, so just like with Locke-Volterra predator-prey, with Locke-Volterra competition models, we're going to look at isoclines. We can't really integrate these models like we did with single population models. There's no closed form solution. So the way we understand the dynamics, the way we understand what happens in these systems when we have multiple different equations is we look at these isoclines. And again, isoclines are where the populations stop changing. So they're zero population growth isoclines. The population doesn't move anymore, it doesn't change, and basically that means we're going to set these equations equal to 0. So they're conditions where the population size of species 1 does not increase or decrease in size for any combination of N1 or N2 that lies on the isocline. So that definition is not super insightful. I think working through what these isoclines say and what they do is much more insightful than that particular definition. So if we want to find the species 1 isocline, just like with the predator-prey model, if we want to find the species 1 isocline, we set the species 1 equation to 0, right? When we want to find the isocline for a particular species, we set that species equation to 0. So here I've got the equation for species 1. I set it to 0. That's going to give me the species 1 isocline. And again, that means that the rate of change of species 1 with time is 0. Species 1 is not changing through time. Whenever we're on that isocline, whenever we're on that line, the species 1 population does not change in time. So I can divide away the R1 and the N1. They disappear, and so all I'm left with is that bracket. And so solving for that bracket, that bracket basically gives me the species 1 isocline. What I'm going to do is multiply through by K1 and solve for N1 just to get that equation. And this tells me the relationship between species 1, the abundance of species 1, and the abundance of species 2 that I have to have satisfied to be on the species 1 isocline, to be at a point where the rate of change of the species 1 population is 0, where the species 1 population is not changing. Let's take a look at what that looks like. So here I am plotting the abundance of species 1 on my x-axis and the abundance of species 2 on my y-axis. My choice of axes is completely arbitrary. For now, I'm just going to put 1 along the x and 2 along the y. All right, so now I want to plot this line. This is exactly like plotting y equals mx plus b in high school. You can plot it any way you want, but the easiest way to plot it is probably to find the intercepts on the two axes. So let's take a look. There's our line. Let's look at where it should be crossing. If I look at the intercept along the x-axis, right, along the species 1 axis, that is basically where my N2 is 0. I'm at 0, you'll notice, along the y-axis. And so N2 is 0, and that means I want to stick N2 in as 0, so that term disappears. And that tells me that the intercept along the x-axis is at K1, or the carrying capacity of species 1. Good to go. I've got the one intercept. What's the other intercept? Well, if I'm looking at the other intercept, it's the same line, but this time I am looking along the x equals 0, along the y-axis, right? I'm looking for the intercept along the y-axis, and so that means my x, or my species 1 abundance, has to be 0. So if I put n1 equals 0 into the equation, now I can solve for n2, and I find that n2 is equal to the carrying capacity of species 1 over that competition coefficient that describes sort of the impact of species 2 on species 1. All right, so that gives me the isocline. Another way of thinking of this isocline is sort of how much species 2 substitutes for species 1. So if I have no species 2, that is my intercept along the species 1 axis. That tells me, how many total species 1 can I support, right? And so not surprisingly, that intercept is the carrying capacity. But then as I add more and more species 2, as I move along the y-axis higher and higher up, I can support less and less species 1 because species 2 is stealing some of the resources, right? So if I have no species 2, I can support my full carrying capacity of species 1. But as I add species 2, as I move up along the y-axis, I get less and less species 1 until I hit a point where I can't support any species 1 anymore, and that's my intercept along the y-axis, right? That's where my species 1 is 0. And that point where I can no longer support any more species 1 because I have just too many species 2, that is basically the carrying capacity of species 1 divided by that competition coefficient. So again, that's another way of thinking of isoclines. Now again, if we think about the isocline as what the system can support, if I am below the isocline on the left, so we're looking at, again, this is species 1 isocline, so if we're understanding which direction the trajectories are going to move, we have to look to the left and the right along the x-axis because that's the species 1 axis. So the species 1 isocline goes with the species 1 axis, so we're looking left and right along the x-axis. On the left, I have fewer of species 1 than I do at the isocline. And so I have fewer species 1 than the system can support. And so as you might expect, on the other side, I have more species 1 than the system can support. And right along the isocline, I have exactly the number of species 1 that the system can support for any given amount of species 2. So if I have fewer species 1 than the system can support, you would expect my species 1 population to increase. And so that means I'm going to move to the right along the x-axis, because again, the x-axis is the abundance of species 1. If it's increasing, it's moving to the right. On the other side of the isocline, I have more of species 1 than the system can support, and so in this case, I should move to the left to fewer species 1, along smaller numbers on the x-axis. All right, so that's going to become important in the next class because you're going to need to know how to draw these. For this class, you just need to sort of understand that. But in the next lecture, we'll talk about why these arrows matter. All right, so that's your isocline. Now let's think about the species 2 isocline. And again, to find the species 2 isocline, I want to set the species 2 equation to 0. Right? If I want to find the isocline for any particular thing, the species 2 isocline, I want to set its equation to 0. If I do that, again, that tells me that the rate of change of species 2, the rate of change of the species 2 population through time is 0. It's not changing. And again, I can divide out the R2 and the N2, and that just leaves me with a bracket. And that bracket, basically, when I set that equal to 0, that defines the species 2 isocline. So again, I'm going to multiply through by K2, and I'm going to solve for N2, and that's going to give me this equation. So once again, I'm just going to plot this equation. It's just plotting a straight line. The easiest way to plot a straight line is to find the intercepts along the x and y axis, or in this case, the n1 axis and the n2 axis. And once again, I put the n1 axis along the x and the n2 axis along the y. That's somewhat arbitrary, but that's what I've chosen to do. All right, so here's my species 2 isocline. Let's figure out what those intercepts are. So if I take a look at the y-intercept, right, that is where my x is 0. That's where my n1, because n1 is along my x-axis, I'm going to set n1 equals to 0. So if I set n1 equals to 0, that term disappears, and that tells me that I'm intercepting that y-axis. I'm intercepting along the n2 axis at n2 equals k. So that gives me the y-intercept for the species 2 isocline. Now let's take a look, go back to that equation for the isocline. It's the same exact equation, but let's figure out where it intercepts the x-axis, where it intercepts the n1 axis. So it's going to intercept the n1 axis where the y-axis is 0, where n2 equals 0. So if I put n2 in as 0, then I solve for n1, and I find that n1 equals the carrying capacity of species 2 divided by that competition coefficient that describes the effect of species 1 on species 2, the competitive effect of species 1 on species 2. And again, we can think about this species 2 isocline as describing the sort of impact of species 1 on species 2, or the trade between how many individuals of species 2 I can support for more and more species 1. So if I have no species 1 in my population, then I can support the full carrying capacity of species 2. So it's not surprising that that y-axis intercept is the carrying capacity. However, as I add more and more and more species 1 to the system, I can support less and less and less species 2, right? And so that line decreases, I get less and less species 2, I get lower and lower along the y-axis, the more I move along the x-axis. The more species 1 I add, the less species 2 I can have, until I reach a point where I have so many species 1 that I can't support any species 2. Species 1 is eating up all of the food for species 2, and as a result, I'm out of luck. And so that point is the carrying capacity of species 2 over that competition coefficient. All right, so again, when we think about that isocline as being sort of a marker of what the system can support, this time we're looking at the species 2 isocline. So we need to think about the species 2 axis. We need to think about the vertical axis, the y-axis. And so on this side of the isocline, I'm lower on the y-axis. And so that means I have fewer species 2 than what the system can support. On the other side, I'm above the isocline. I'm higher up along the y-axis. And so I actually have more of species 2 than the system can support. When I have fewer species 2 than the system can support, then I would expect my abundance of species 2 to increase. And increasing, because we're thinking about the y-axis, we're thinking about species 2, means moving up. When I have more of species 2 than the system can support, you would expect species 2 to decrease. And because we're thinking about the y-axis, we're thinking about species 2, that means moving down, moving vertically down. And so again, we're going to think about these arrows more in the next class, but right now I'm just describing what happens to the species 2 population on either side of the species 2 isocline. That's going to become important for the next lecture. So ultimately, when I have two different species, there's 4 possible ways I can combine isoclines. I can draw the isoclines like this, where the species 2 isocline lies entirely above the species 1 isocline. The 2 is shown in blue, the 1 is shown in red. I can flip-flop those, so I can have the species 1 isocline lie entirely above the species 2 isocline. Again, that's the red one lying entirely above the blue one. And then I can have the crisscross scenarios. So for the one crisscross scenario, I have the species 1 isocline lying above the species 2 isocline on the species 2 axis, but below the species 2 isocline on the species 1 axis. So again, in this case, the isocline that is higher on each axis is the isocline that is not the same as the axis. The species 1 isocline is higher on the species 2 axis, and the species 2 isocline is higher on the species 1 axis. I can flip-flop that again, and I can have the species 2 isocline above the species 1 isocline on the species 2 axis, and the species 1 isocline above the species 2 isocline on the species 1 axis. So in this case, the isocline that is highest on each axis is the isocline for that axis. Again, in the next class, we're going to dissect this. We're going to look at the different trajectories, the ways species 1 population moves on either side of the species 1 isocline, the way that the species 2 populations move on either side of the species 2 isocline, and that's going to tell us what's going to happen in each of these different scenarios, in each of these 4 different ways that the isoclines can intersect. For today, we're just going to look at trying to draw those isoclines, trying to find those intercepts along the axes, and plot out what they look like for 2 different species to know which of those 4 different scenarios we're falling into. So these are going to be worked out examples in class. If you aren't in class, I'd encourage you to try to draw these isoclines and make sure you know how to do it so that you can do it on a test. And again, this is going to be very important. We're going to need this. We're going to need to be able to draw these isoclines to figure out what happens in these systems. So this is an example, again, with needle ants, but in this case, looking at competition with winter ants. And I'd encourage you to do that on your own. And if you can't, I'd encourage you to get someone else's notes. And then we're going to do a second worked out example looking at blue jays and crows. Again, plotting the different isoclines, trying to figure out what those isoclines look like and which of those 4 different scenarios we are for how the 2 isoclines can be positioned relative to one another. All right, if you have any questions about competition or about isoclines or about drawing the isoclines, figuring out where those isoclines are on the 2 different axes and which of those 4 scenarios we're in terms of how the isoclines are relative to each other for 2 species, I'd encourage you to email me and I will answer those questions. Otherwise, I will see you next class where we'll look at what these isoclines mean and how we can use them to predict the outcome of competition.